Well, thanks everyone for coming. We have a great hour here to talk really deeply about some things that I'm really passionate about with regard to environmental DNA and looking at how we can use environmental DNA, things that are shed from species as they move through their environment to understand community structure and really the health of ecosystems. And as we are thinking about these things today, I want all of you who are in here just to be as engaged as possible. I'm going to go through a bunch of technical material, but I think one of the things that surprised me as we've been talking about how we're using this genetic information that we're pulling from ocean water or lakes and rivers to inform all kinds of different types of decision making and all kinds of different natural assessments is how accessible it is to so many people who are working in different domains. Somewhere I have a clicker here. It's my job. So my name is Peter Thielen. I am a senior scientist at the Johns Hopkins Applied Physics Laboratory. We are a special part of Johns Hopkins that is on a separate campus. We sit halfway between Baltimore and Washington, D.C., and a large part of the work that goes on within the Applied Physics Lab is facing the US government and doing things that often end up out in ocean systems. There are so many different systems that we need to care about when we're putting ships out into these environments and when we are really trying to push technologies in various directions. And my work as a molecular biologist in this domain has really focused on using genetic information so that we can understand the world around us. That genetic information that you might be familiar with now from reading the newspaper and looking at what the most recent COVID variant is. That is a piece of genetic information that is directly informing something in your daily life. And the same principles can be applied essentially to how things are floating around in the ocean and how we assess those ecosystems. Within our work at the Johns Hopkins Applied Physics Laboratory, we have focused within my department on problems that look like they're 10 or 20 years out. And I've now been able to be engaged in this domain for long enough that what looked like it was 10 years out 10 years ago is today. And it's been a really exciting time to see all of these things mature as technologies and then be pushed into various national strategies and different activities. Chris Meyer is one of my close collaborators, and unfortunately he was unable to make it here today. He is at the Smithsonian National Museum of Natural History and has an amazing collection that I'll talk about in a bit. And really, as we have worked together, both between an academic institution and a government institution, that transition of new technologies to standard practice and using the best available science has really been catalyzed through these types of engagements. I'll also say he has maybe the coolest title, curator of mollusks. And as we get this conversation going, I want to just do a brief around the room and get some hands. Like, do any of you work in R&D? Great. Do any of you work in the ocean domain? A few, awesome. Did we get any government or regulatory folks in the room today, either from local or state or federal side? Great. Across all of these, right, energy industry as well, what we're seeing is a vast array of applications as people are looking to more efficiently characterize the natural world around them. This can mean everything from the really basic research that we do all the way to much more dramatic implications that Chris and others are interested in. And today we're going to talk about really deeply what environmental DNA is and what that means to myself and how it can be extended to all kinds of other applications. What's going on in this landscape at the moment? I think it often falls by the wayside that there are hundreds and hundreds of scientists around the country in the United States and thousands around the world that are pushing really intensively on development of these technologies so that we can use them for essentially digitizing our biological records. And then as we look ahead, I want to get into some discussion today with all of you on how we can bring these technologies forward and not develop them in small pockets all over, but develop them in a way that allows us to expedite the process and really make the most out of this. So one observation that I have had sitting from my position as a basic research scientist is that as we look into the environment, right, when you walked into the building today and when you go outside, the technologies that exist to understand what species are present are very limited, right? So we use our ears and our eyes to look for species. We remember those things, but then that goes away, right? That is a very short-lived piece of information. And these technology improvements that relate to environmental security are myriad. I think the most recent numbers that I'm able to pull regarding just the blue economy, things that are focused on the ocean, is every year, just within the United States, there's about $400 billion of revenues that support 20 to 25 million jobs. And as we look at what we're monitoring for protected species and invasive species and all of these other applications, there have been some general truths that I've been able to identify through all of the different groups that we've engaged with. One, whether you are an environmentalist or you're working in industry that is leveraging oil and gas platforms or wind platforms or any other application, it's in nobody's best interest to harm protected or endangered species. There is not a single person that wants that to happen. Oftentimes, the conversation gets going when we are talking about what that means for broader infrastructure means. The ports that we have and ships coming into our countries to deliver goods and services, organisms bind themselves to the hulls of ships, and that slows them down, and that causes their emissions to increase, and it causes all kinds of cascading effects. As those things are carried on the hulls of ships and in ballast water, and they are just part of the commercial process of moving goods and services from point A to point B, our ability to make all of these ports and all of the infrastructure that exists as efficient as possible is in everyone's best interest so that we can maximize productivity, reduce costs, and really drive all of the efficiencies as high as possible to take advantage of the capabilities that we have. With regard to the things that are attaching themselves to these ships, invasive species are another element that are drastically underappreciated within the United States. I personally grew up in Minnesota and had a family that was in the part of Minnesota that is right on Lake Superior in the northern part of the state. There's a huge industry up there bringing iron ore back and forth between the ocean and through the Great Lakes. And as that's happening, one of the major invasive species in the United States that some of you in the room might be familiar with is zebra mussel. Zebra mussels are incredibly small. They hitch a ride on almost everything. They spawn, and their larvae can be carried in just the water that goes into boats that have been in these bodies of water. They can survive in saltwater and freshwater, right? And they clog intakes, they clog ports that are being used for cooling, and they cause essentially billions of dollars of damage a year. And our ability to counter these invasive species and prevent them from invading in the first place is a really essential part of how we understand these biological communities and how we push ourselves to not only have to reduce all of the activities that go into ridding ourselves of these invasive species, or even attempting to, but spreading them from one location to another. And there are a lot of applications that folks are working in the invasive species domain with regard to environmental DNA. And the last is really natural resource assessments. Right now, when we are counting the number of fish in the ocean, we are sending ships out on regular occasion, and we are catching them with nets, and we are counting them, and we are doing this with some frequency so that we can have an understanding of how many fish and of what type are in the ocean. Those fish assessments require a ship. This is it might seem self-explanatory to folks, but it's difficult to overestimate how expensive it is to support those ships. And when a ship needs to be replaced over time, those also are getting far more expensive as well. So these are very niche applications that are required so that we can manage our natural resources, and those natural resources are used to literally put food on your table, right? It's not just we want to know how many fish there are and what type. These are the rules and regulations that are put into place so that we actually can take things out of the ocean that have both a benefit to you as a consumer as well as the groups that are using these natural resources for their day-to-day profit. I've touched briefly on how we're using environmental DNA, just conceptually, right? DNA is in your cells. It is in the cell of every fish, the cell of every marine mammal. It is in jellyfish. Everything that's alive has DNA within it. That DNA can be observed using modern biotechnology and things akin essentially to a COVID test in some instances. And those genetic signatures can be shed from each of these organisms into the environment, and using standard biotechnology tools, we can then extract that information out of a cup of water. So the species that are living in the sediment on the bottom of the ocean, whether they be oysters or clams, things that are living in the water column like fish or marine mammals, or things that are above the water. There are many protected species, there are many bird species. All of these species essentially can be detected when we're looking at a cup of water. So that genetic information is shed from that organism, it spreads within the environment, and then we'll take a sample, we'll filter that down, and use the biological components of that essentially to extract that information out and then make sense of that on a scale that is invisible to the naked eye, right? But ends up being a conversion from something that is chemical, that's floating around in water, to something that's durable and that is easy to record and then look at over long periods of time. So as all of these technologies have been coming together, it's folks like those of you who are in this room oftentimes that are beginning to realize that this ability to discern species within a complex sample is technically possible, right? We've had the great luxury, I will say, and the great ability to work with people across the country and across the world and push these technologies really to their limits, right? So as we're developing these technologies, we're not just thinking about what we can do in the long term. We're thinking about how do we transition these from a research capability that exists in the laboratory today to something that we can do out in the environment. This is an image taken in Antarctica where we were taking water samples at depth and correlating these with physical oceanography measurements. And essentially, we, as our group at Johns Hopkins Applied Physics Laboratory, have been able to go to both poles and go to both the Arctic and the high northern latitudes and the Antarctic and the high southern latitudes and look at how we're able to deploy these technologies and what type of information we're able to get from these pristine environments. On a personal level, this has been really transformative because oftentimes as a scientist, we get kind of legitimately stuck in the lab and we're writing proposals and we are writing reports and doing all of these things, but actually being able to go out and do these types of field activities has been a great experience. And I also think that it's hard for people to really understand from a picture, right? So one of the elements from this Antarctica trip that I like to share with folks is our last day when we were on the western side of the Antarctic Peninsula. We're on an ecotourism ship, and that ecotourism ship was going to take passengers and drop them off to see some penguins. And we had seen whales on the way in to this fjord, right? And it was beautiful. There's glaciers on both sides. And as we went in, we had made a plan to then take a small inflatable boat and go back out to where the whales were and see if we could find them and collect samples in their proximity to see if we could identify what kind of whales are from the environmental DNA that's in the water. And we had seen these whales, and we had even been able to hear them on the way in because they were so close to the ship that we were on. But then when we got out there on this boat, we were surrounded by icebergs and things that had fallen off of these glaciers, and we are trying to navigate through all of this, and there's the rumble of the engine, and we were unable to find these whales, and one of the people on this boat just asked a simple question: what if we shut off the engine? Right? And it went from this just low-level noise that we hadn't been bothered by because that's what we were out there to do, right? We were taking a boat to go see where these whales were. And then all of a sudden, we had this perfect silence where we could hear the glaciers rumbling around us and the ice cracking, and we could hear the water splashing on the icebergs. And after 10 or 15 seconds, we could hear off in the distance, and these whales that were just out of sight that were all around us, and we could hear, 360 degrees that there were whales that were present. And this moment where we're out there trying to collect a sample, and we have the noise and the activity, and we're in this pristine environment, is one where, it's really transformative to be able to think that this is something that we can introduce as a technology when people are there on ecotourism ships to collect samples and preserve that in a digital form, rather than have to rely on the experiences that very few of us are going to be able to have throughout our lives. As we've been on these ecotourism ships, first, they're incredibly welcoming to scientists, and we end up essentially educating those who are going to these locations. And we have essentially pushed everything that we do in the lab to minimal capabilities that can be deployed on a ship like this. We can put together an entire genomics lab using commercial off-the-shelf components and really highly customized molecular biology and software solutions for under $10,000. And that might sound like a lot of money for those that are not used to putting together custom genomics capabilities, but the fact is these are able to essentially run off a typical laptop. They fit in your pocket, the sequencer itself, and they're incredibly inexpensive to operate when compared to a typical brick and mortar laboratory. As we've been pulling all of these components together and going to these different places, one of the things that's obvious, though, is that this is not just us taking a cup of water out and saying, oh, look at all those organisms that we found, right? There are probably 2 or 3 dozen steps that are hands-on that take somebody like myself with their doctorate, or any of the trainees, or any of the other people that are working in this domain, that are going from that point of collecting a water sample to a piece of data. And our goal within our group has really been to push this process as hard as we can from the technical side so that we can go from sample to analysis in a way that does not require the same amount of expertise, right? And this amount of development and amount of research and prototyping and all of these different components that are required here really requires kind of an all hands on deck. And I think the other conversation point that we can get to, when we talk about sample to answer, that answer is going to be different for every single person in this room. It's going to be different for different applications that we have on the ship. And it means a lot of different things to extract that information out and use that as that durable resource that is that permanent data that we can archive. So our approach to doing this has been really modularizing our processes so that we're able to go from that water sample to that final piece of data as quickly as possible. So we've developed a number of capabilities that are as simple as putting a few things in a Pelican case so that we can push a button after you put a filter on the end of a tube and dip it in the water, and then you take that sample back with you to a lab that's prepared to process it through the entire multi-step process. We also have been developing a microfluidics capability that takes all of the reagents that we would normally pipette from point A to point Z through those protocols and freeze drying them on these chips so that they can last in a way that's shelf stable. We don't have to rely on the long cold chain resource, and really get to a point where we don't require the same amount of hands-on with the idea that eventually this could be a fully autonomous in situ analysis capability to go from that water sample to a piece of information that comes out and not have to have that person present. And last, one of the things that we've deployed in multiple different form factors is essentially an edge compute capability that uses an NVIDIA chipset. And the same tools that you use in video analysis or things that require analysis on site so that you can minimize that data package as much as possible. And rather than distributing tens or hundreds of gigabytes of A's, C's, T's, and G's to then go be analyzed with some other high-performance compute clusters, we can actually minimize the computation requirements so that they work on these edge compute devices and are able to fit into a really small Pelican case and essentially analyze data from those tens to hundreds of gigabytes down to a few kilobytes and then be transferred over a satellite uplink. So as all of these capabilities are coming together, right, we're developing and we're putting prototypes out into these environments and we're breaking things as much as we can. It still requires us to go out there and do this work in this process. What's become very apparent, though, is that elements of this can be branched off and used immediately. And this is where our collaborations with Smithsonian and other groups around the country have really emphasized the readiness state of environmental DNA work. Chris Meyer, who is in the middle image all the way off on the right there, has been working with folks all around the world to collect samples that they can then bring back to their laboratories, and they can analyze those samples in a way that gives them an idea of what organisms exist within the environments. This is in the South Pacific somewhere or other. They were just there for about a month. They started in New Zealand for some work and then did some other activities. What you'll notice here is they're actually in the bottom right image using just a regular 50 milliliter syringe, right? They're taking water and they're pushing it through a filter, and that is sufficient. There's no electronics involved, there's no expensive hardware, and that's basically what's required to collect a sample in these environments. What we've been able to push further with them is taking these little Pelican cases that we build, putting these pumps in them, putting multiple pumps in them so that they can just increase their throughput, right? And it goes from being hard to push 50 milliliters of water to now you have a small machine that's pulling that through at a liter every few minutes, essentially. And it gives you the ability to collect much higher numbers of samples and higher volumes. And basically that filter that's on the right there is one example of a final sample that's ready to go into storage and then go to the laboratory for that kind of analysis. And Chris and his team have gone all around the world and collected samples as they're undertaking their research and the work that they do at the National Museum of Natural History. If any of you get a chance to get to DC, the behind the scenes of this collection is one of the most amazing experiences that I think is out there to experience. Chris and the rest of the team that are working at the National Museum of Natural History, in addition to having a beautiful genomics lab and beautiful genomics capability, they have a collection of mollusks that is from the entire world that represents every single species that is known as far as we are aware. I have never seen shells like this, and they are some of the most pristine, beautiful examples that exist, and these are in national collections that are there to serve as resources. And it's one thing to say, we have a giant shell collection. It's another thing to recognize that within every one of these shells, as they're cleaned, essentially there are parts of those animals that are left behind in the shell. Those parts of those animals can then be recovered, and we can sequence parts of their genomes, and they then serve as voucher specimens for all of the other species that are within that species genus or even further defined. So beautiful collection, highly recommend seeing some of the things that the Natural History Museum has to offer in this domain. And beyond shellfish and these voucher specimens, the other element that I think is really important is that any collection that can exist in a form that is physical that is a biological specimen can exist in a digital format as well. And this conversion from these physical collections to these digital collections and our understanding of the genetic sequence of these organisms has dramatic implications for how we manage our natural resources. Right now, the Natural History Museum is undertaking a large project to sequence the genomes of all fishes within North America that exist in lakes, rivers, and oceans. And by generating the genetic resources for all of these species, this then serves as a durable resource for us to understand the dynamic nature of species as they exist in their environment and really look more broadly at these sustainability topics that a lot of us care so deeply about. So as we look ahead, and I want to get into some conversation here, I just want to give some updates on what's going on in this community. So there is a huge groundswell of effort by scientists across academia, industry, government that have been coming together now for going on 7 years that recently joined Chris and myself at Johns Hopkins Applied Physics Lab, as well as the Smithsonian, to really convene around this topic of how do we do this with environmental DNA in a coordinated way that doesn't require us to do all of the development in the little pockets that tend to exist, and how do we push these capabilities in a manner that supports strategic efforts. So within the federal government last year, there was a strategic document that came out that was announced at this event. And essentially, this was consensus from people across many different domains of how we would implement environmental DNA science as a best available method to assess these natural resources that we have, and how do we do this in a manner that is cost-effective, that is recognizing the myriad needs as we're looking across all of our different domains, and really engaging as much as possible. And I think this is especially important as we look more broadly towards all the different applications, is how are we going to do this in a way that's sustainable, and how are we going to do this in a way that we communicate out our findings and the capabilities and leverage all of the groups around the world that are doing this, not just those of us who are working on this in the United States. So we have started coordinating, and all of those that were involved in the planning of these previous events have now been working under the auspices of the Marine Technology Society. And Marine Technology Society as an organization is great at getting people together and hosting conferences and workshops and expressing diverse viewpoints. Marine is not the only element of this, right? And so even the strategy that came out last year for environmental DNA got renamed at a certain point from the marine environmental DNA strategy to the aquatic environmental DNA strategy. And I think that's a testament to how broad the applications are here. And I'll have a QR code up at the end as well for those of you who want more information there. And so as we start to get into conversation, I want to place a couple of other thoughts. I started with this thought that dramatic technology improvements are required for us to advance environmental security. Environmental security means a lot of things to a lot of people. And as we're talking about environmental DNA, it is a way for us to physically capture what was present in that sample and archive that in a digital form, right? That is one element of how we can observe these natural systems and how things are changing and all of the different components of that environment that we care about protecting in many different ways. Other things that might not quite be on people's radar are, there's a NASA capability that went into orbit last year that is essentially looking at ocean color for phytoplankton and the things that are existing in the water. What they don't necessarily have is access to the water itself, right? They can see the water, and they have coarse resolution over the entire ocean with high frequency, and they can see how these things are changing over time. But the ability to actually extrapolate between a sample that's taken in one of these locations and those images that are being collected requires a lot of intermediate studies so that we can have high confidence. in all of the dynamic changes that happen every day in the ocean. Another component that a lot of people are going to be familiar with are things like whale sounds. And a lot of effort has gone into really deciphering these audio recordings and getting information out of them that adds additional layers to all of the different species that we care about. And so when we're talking about environmental DNA monitoring, this is not the only way to do things, but it has specific advantages that are durable, that allow us to collect and archive samples. But the ultimate dream for a lot of us who are working in this community is to connect these with hyperspectral systems, space-based assets, connect with hearing, right? The things that we can listen for in the ocean. And so this ends up being something that you can see, something that you can touch, and something that you can hear, right? And right now, because we don't have that ability in the middle to touch the thing that's present unless we are physically there and have a recording of that, this really fills out a picture of how we can better assess these environments and how we can dramatically push our capabilities in this field. So I want to sit on this slide for a minute because I think, as people have been thinking about this with respect to the things that you do every day, the things that you care about, whether it's the natural resources that are taken out of the ocean, right, from fishing vessels or things that you consume on a daily basis from the ocean, whether it be oil, gas, and wind installations that are already existing or going to exist, whether it be in that third panel here is a hydroelectric dam, right? If these hydroelectric dams need to be cleared out frequently because of invasives or they stop producing in some way, shape, or form, or we're talking about installations where we have lots of ships that cost a lot of money and provide a lot of services that we depend upon, right? How does this impact you? And I think really this has been a discussion for a lot of us within this community that cares about sustainability and applying new technologies. And I want to give some contact information for those that want to keep in touch. And I think one of the most important things is we're at this place right now is bringing recognition to the fact that there are technologies out there that allow us to observe the environment in ways that have not been previously possible. They're cost effective, the technology is ready, and the things that we recognize we need at the moment are really this groundswell of both the scientific community and those who are in a position to leverage this data for commercial needs, for conservation needs, for any other application, and get these technologies into the hands of the people who need to go from that raw sample that's in the environment to a piece of information that they can make use of over the longer term. So I appreciate everybody's attention today and look forward to being able to catch up on a couple of side conversations afterwards. So thank you.